Hi everyone.
Bro.
So today I'm going to give a quick introduction on high throughput screening. So first, what is HTS? HTS is basically an automated research method designed to run large volumes of biological or chemical tests in a short time. It's often employed in pharmaceutical and biotechnology industries for drug development and also in academia for research purposes. And then the primary goal of HTS is basically to identify active molecules called hits or leads, which are basically candidates that affect targets in a desired way. This could be we want them to modulate certain biological pathways, interact with proteins, or inhibit certain disease-causing targets. However, it's important to note that HTS does not identify drugs. Instead, it simply finds starting points for drug design. And this is because it can't evaluate toxicity, bioavailability, or other factors that are necessary for a successful drug. So we can basically understand HTS to be a fast scan of biological processes by which compounds with little or no effect can be rapidly excluded from the drug design pipeline. To go over how HTS works, you first start with preparing the samples and compound libraries. Samples are arranged on microplates that can be typically in a 384, 1536, or so on well formats. And then compounds are stored in stock plates and transferred to assay plates for testing using automated pipetting systems. For the compound libraries, you can choose different types depending on what you are looking for. Certain ones include diversity-oriented libraries, which kind of have a broad chemical diversity to explore new potential interactions. You could also use a focus library, which basically contains compounds with known activity against similar targets to see if there's also an effect with the target in question. And another option would be to focus on natural product libraries. So this contains compounds that are derived from plants, bacteria, fungi, or other natural products. Next is to establish an automation method. So automation optimizes HTS by minimizing reagent use, reducing manual intervention, and ensuring assays run in single wells. After that, you would have to configure a robotic workstation. So a robotic workstation basically allows you to autonomously manage multiple plates simultaneously, which speeds up data acquisition by a lot. The robot is responsible for managing microplates from station to station for several steps, including reagent addition, mixing, incubation, and detection. And then last step, once you've finished all of that, you would have to acquire your data and analyze it. So data in HDS is usually collected via optical methods that quantify the amount of light produced by the sample. You could also, in certain special cases, have specialized instrumentation like the multi-mode microplate readers, which can sequentially perform different experiments or apply different detection protocols on the wells. And then for this specific instrument, the output would be a grid of numeric values. And then for a brief summary of the strengths and drawbacks of this method, I think the biggest strength would be speed. So HDS platform with 1,536 well microplate can handle over 200,000 assays per day. This is a big jump from if you were to do it manually. And then another strength would be automation. So because it's fully automated, the workflow minimizes human involvement, which in turn improves precision and accuracy because it removes variability introduced by the manual handling. The last strength that I mentioned here is scalability. Because it's possible to screen massive chemical libraries with an automated system, we can kind of go a lot faster with the different chemicals involved. And there's also reduced reagent costs because you don't need that much per reagent. So overall, your costs will be lower. For the drawbacks, a big one in the beginning is the cost. So initial setup for HDI is very expensive because you need a lot of specialized equipment. And then because it's such specialized equipment, you also need highly trained specialists to operate it. So there's also personnel costs to consider. One thing that HTS is kind of known for is having the potential to generate false positives, which are basically compounds that only appear active in the first run. And this necessitates secondary screenings, which obviously would cost more and also take more time. And then finally, there's also certain target limitations. So HTS is very effective for well-characterized biological targets, but less so for more poorly understood biological pathways. Then lastly, just to go over the development timeline really quickly, starting in the 1970s to 80s, there was very rudimentary early manual screening methods. So they were very time consuming and only allowed for testing of a few hundred compounds. And there were very rudimentary robotic systems. Fast forward to the 1990s, there was an advancement in robotics, liquid handling, and miniaturization. So this allowed more automation to come, and pharmaceutical companies could now adopt HTS to screen for compound libraries, drug discovery. There's also the introduction of multi-well microplates. So this enabled large-scale assays using tiny volumes, which again would reduce costs and speed it up. And then the major technological advancement of this time is the fluorescence-based assays, which allowed for faster and more sensitive readouts. And then in the 2000s, cell-based HTS was developed, which enabled screens to mimic more realistic biological environments by using live cells to assess compound activity. And then it was also kind of gaining traction during this time, and it was increasingly adopted by academic researchers, biotech companies, and certain government labs. And then finally, for the present, we're currently able to use ultra high throughput screening, which is capable of screening millions of compounds in a matter of a few days. And yeah, that's a basic quick introduction of HTS. Thank you.
All right, so today I'll be walking everyone through this paper called High-Throughput Microbial Culturomics using automation and machine learning. This is a paper by some of the postdocs at the lab that I'm currently working with, Harris Long's lab. And their system called CAMI is a very cool robotics platform that I've used a lot for my culturomics projects. So I'll talk about that today. First, I just wanted to quickly introduce everyone to the concept of culturomics. It's a sandwich word between culture and omics, and really it's the process of isolating and studying bacteria in culture to better understand their roles in microbial ecosystems. Traditionally, isolating bacteria from environments like the gut has been labor intensive and often yields limited diversity. So this paper introduces a new automated system for streamlining the whole process. So to justify whether why the CAMI platform is a very economically viable choice, there are many challenges to traditional culturomics methods. It's very labor intensive, as I mentioned. Traditional methods, you would pick every single colony manually. It's really time consuming and it requires a lot of human effort, sitting at your bench, picking single colonies for days at a time just to get like a few hundred if you're fast. There's a very low diversity of isolates. Traditional approaches tend to repeatedly isolate dominant bacterial strains because by the human eye, we tend to simply go for the easiest picks on a plate, and that might lead to similar strains being picked multiple times. There's limited scalability, as I mentioned, with the time constraints. Manual techniques cannot handle the large number of colonies needed for comprehensive studies. So again, it's also not randomized because the human mind will inadvertently shift towards certain picks. So and it's also so expensive. So this is why we have the CAMI platform. This is a robotic system essentially that takes in a plate that has a mix of different bacterial samples and then uses an algorithm to identify the most interesting or different or morphologically diverse colonies within a plate of mixed bacteria, and then it picks them and then isolates each single strain of bacteria into a 384 well plate. So just to go through some of the quick procedures of this entire platform, we start in an anaerobic chamber, and this is because some of the work that we've done previously is with gut microbiome, so it's obviously an anaerobic condition. So you pass the plates in through port number 13, which is illustrated on the graph. And then you'll have this robotic arm, which is number 16, that stores all your plates or samples on the racks, which is numbered by 15. So the first thing that the arm does is it places the racks on what number 9 is, which is like a desealer and sealer. So it rips off the seal on the plates. Then the arm transfers the plates to number 17, which is called Hudson, I think, micropick. the Hudson Rapid Pick MP colony picker. This is like the most important part of the system. And really what it does is just take the samples that have been machine learning identified already that are most interesting, diverse, whatever, and then it picks them and then inoculates them in 384 well isolates. So yeah, this is basically how that system works. And before I get into like the results of all this, I just first wanted to give like a quick aside, which is machine learning. So Steven knows a lot about this, I'm sure, but I'm just gonna give you the simple version. So machine learning, especially deep learning, involves training a model to recognize patterns in data. This is the overarching goal of machine learning. As shown in this image, the process starts by feeding the model a set of training data. So in this case, original images of colonies. So if you have like, if you have like one rectangular plate and there's like a bunch of different types of bacteria on it, and it's like messy growth with colonies touching each other and whatnot. We humans have previously annotated the colonies to represent certain phylogenetic categorizations. So once you feed the machine this data, it will learn how like the specific relations between like these sets of training data. So you have differences in like thickness or absorption, fluorescence, whatever dimension really. And then it learns it and develops the ability to automatically classify and segment new images based on similar patterns. So for the CAMI platform, this deep learning process is applied to colony morphology, right? The system is trained to recognize various colony features such as size, shape, color, and texture so that it can autonomously pick the most morphologically distinct colonies by those criteria, right? And then this makes the isolation process more efficient and also develops a more diverse selection of picks. So yeah, also there's a lot of different applications for machine learning, which I won't go into. So this is specifically how the machine learning algorithm works. You have scaling and dimensionality reduction. So really what happens here is you process the images and measure the characteristics, like I mentioned already, and then you do principal component analysis, which is basically deciding which of those criteria is the most important. And then you do a demultiplexing, which is just reducing multidimensional space. So say you have 4 dimensions: size, shape, color, and fluorescence, right? So that's like a 4D space, basically. That's like you have different measurements for these 4 different things, and it doesn't really translate well to like graphs. So how can we do so how can we reduce that, right? We do PCA, we reduce the dimensions of the matrix so that it's analyzable by a machine. And then you do your algorithms so that the scaled data is embedded into a multidimensional space where colonies are represented based on their morphological features. And then you optimize the algorithm to find the colonies that are most distant from each other within the 4D space, right? It's kind of hard to imagine. You're not supposed to be able to imagine it, but the algorithm is able to find the 2 most distant points, essentially, within that space. And that's how, and you also have this random forest model for taxonomic prediction, which is really helpful because then you can say, okay, I think these 2, I can not only say that these 2 colonies are really distant from each other in terms of their presentation, but I can also say, okay, maybe this is, Protobacter and something else is Pseudomonas. So that's also really helpful because we can link that to 16S ribosomal RNA sequencing. 16S is basically like your government tax ID for bacteria, right? 16S tells you basically what more genus, I think it's genus level taxonomic identity each colony has. So that's also very helpful. So yeah, this is CAMI in action. As I've talked about already, you first take a picture of the plate, And then you can see a bunch of different colonies all looking like different, all presenting themselves differently essentially. And then you run the algorithm. So it recognizes like all the colonies, and then it does its analysis. And then it generates a table of coordinates. So let's say we want to pick the colony in the middle, and then it will just provide the specific like coordinates for the picker to pick it. And then I'll play the video now. Hopefully the volume goes through. I think it's pretty interesting. Yeah, so what you can see here, you see the little red, like really hot, glowing hot thing in the back. That's actually your sterilizer. So all these little needles, they poke on the plate back there first on the media plate. So that extracts your bacterial sample, and then it dips it in media, which are in these 384 well media plates. So that's how you pick a lot of things within a relatively short amount of time. That's basically how it works. That's one Harris one's left. Yeah, I can play it again. Just yeah. And then all these needles are operated pneumatically. So you have, I think, solenoid that actually open and close, and it lets air through. And that's how you get the sort of really fast repeating motion of the pickers to make it faster. Troubleshooting this took a long time, by the way. I helped with the troubleshooting, and the system never wants to work, but it's whatever, we got it working. So, okay. So, all right, so we have some results to share from the paper by leveraging the system of culturomics. So we were able to extract fecal samples from, I think, like 16 humans, and also a few other sources. But over 100K colonies from 20 human microbiome samples, those were fecal. And then out of these, nearly 27,000 colonies were isolated and identified. So this just shows you, like, within the span of a few months, how effective this entire system is, and how efficient it is in generating a huge amount of data related to the identification and storage of different bacteria. So CAMI was able to capture 394 unique bacterial taxa, which means the ML algorithm that I was talking about really works well, because it was able to identify as many different phenotypic presentations of these bacteria as possible. And we were also able to bank all the samples. So whatever has been cultured now has a permanent glycerol storage at -80. And whenever you want to, test something on one type of bacteria, you can just pull it out of the freezer and it will be ready to go. So just a few more words on this image. Figure 2E here shows the results of comparing the gut bacteria found in fecal samples to those isolated and stored in CAMI-generated biobanks to the right. So the left is your identification, and the right is biobanking. On the left, we have a heat map that represents the relative abundance of specific bacterial strains called ASVs found in the original fecal samples. The black bars on the right show whether each strain was successfully cultured and stored in the biobank. So this is another challenge. You saw those needles dip in the well, 384 wells, right? But there's no guarantee that they will actually grow within those wells. And we tried like a few different types of media, like R2A, PDB, and TSA. But sometimes these bacteria, for whatever reason, they refuse to grow, and we don't really have anything to fix that really. But we are still able to culture all the rest, and when they grow, that's how we generate like a biobank, and we can leverage these culture-friendly strains to do further engineering work on them. So what's interesting here is that while the majority of the highly abundant strains were successfully cultured, there was still some abundant species shown in white that we could not culture. These uncultured bacteria represent the current limitations of the culturing method. But overall, it's a pretty good percentage. I think it's something like 70%, right? And then we also have a number of species-specific growth patterns. So in addition to maximizing diversity, CAMI's colony morphology data also helps with targeted isolation of specific bacteria. Cami links the visual appearance of a colony to its taxonomic identity using a random forest model. I talked about that already. So panel 3A shows how different bacterial genera exhibit distinct colony morphologies. So, for example, you have, I think it's Bifidobacterium, which is like the third row, right? Yeah, the third row. So bifidobacterial colonies tend to be larger because as you can see, like the relative heat maps, they look red for the size. While Faecalibacterium colonies are smaller and less dense. So, and then you have panel 3B, which is the real image of colonies versus what Cami predicts them to be, and we can still see that the percentage is pretty good. The random forest model shown in panel 3D uses these features to predict the taxonomic identity of colonies with high precision, around 70%, right? And then finally, we have 3E, which shows the effectiveness of the targeted picking strategy. By using morphology to guide the process, Cami can isolate the desired bacteria much more efficiently than random selection, up to 8 times more efficient for certain genera, right? So this approach not only saves time, but also ensures that we capture the bacteria that we're most interested in without the need to manually screen through thousands and thousands of colonies. So yeah, that is basically this paper. Thanks so much for listening.
Switched out my audio then. Hello. All right. Okay, so I'm gonna give like a pretty quick overview of the HTS industry. So let's first talk about like the market size and the growth. Based on the data, the estimated market value in 2023 is $25.7 billion in general market. And then the projected market value by 2034 will be $95 billion. So the anticipated growth rate from 2024 to 2034 will be about 10.6%. And this will be a plot that kind of shows this trajectory, which is crazy. And then so let's examine like the key segments of the high throughput screening market. Basically, we can break it down into 3 main categories: product, services, technology, and applications. And then so for product services, consumables, it's about like 48% of the market share in 2023. This dominance is also due to their frequent use and replacement in the HTS processes. The software and services are the fastest growing segments here, with the projected CAGR, which stands for consumer annual growth rates of, that's compound annual growth rates of 9.1% through 2032. This growth is driven by increasing demands for data analysis and automation in HTS workflows. The instruments, while not the largest segments, it still remains crucial for HTS operations and continue to see technological advancements. For technology side, cell-based assays dominate the segments, valued at about $12.6 billion in 2022. They're preferring more complex, biologically relevant data compared to traditional biochemical assays. Lab-on-chip technology is gaining traction due to its ability to miniaturize and parallelize screening processes, and also lab-free technology is emerging as a valuable tool for studying biomolecular interactions for labels for reporters. And also ultra high throughput screening represent the cutting edge of HTS, allowing for even faster and more extensive screening capabilities. As for the application side, segment expected to reach about $31.3 billion by 2032. This reflects the critical role of HTS in the pharmaceutical research and development and an ongoing demand for new therapeutics. Biochemical screening remains a fundamental applications of HTS used in various stages of drug discovery process. And life sciences research benefits greatly from HTS technologies, enabling large-scale studies in genomics, proteomics, and other fields, and et cetera, et cetera. So for the key market drivers, we're seeing a significant increase in the research and development (R&D) investments within the pharmaceutical industry. As you can see here, the pie charts, about almost about half of the market shares are coming from the pharmaceutical or biopharmaceutical companies. So basically, companies are allocating more resources to drug discovery and developments, with global R&D spending projected to reach about $254 billion by 2026. Next one would be the rising prevalence of chronic diseases. With non-communicable diseases causing 74% of global deaths annually, there's an urgent need for new innovative treatments. And then CS plays a crucial role in accelerating this drug discovery process to address these health challenges. And then lastly, the technological advancements are revolutionizing this field, and then integration of AI and machine learning with HTS is enhancing the efficiency and accuracy in drug screening. And then for some of the industry trends and innovations, we have the adoption of 3D cell cultures. And the key points for these is that the 3D cell cultures enhances the physiological relevance. It provides more accurate representation of cellular behavior, tissue architecture, and drug responses. For the integration of AI and machine learning, the key aspect will be definitely improves the hit prioritization, and then also enhance the imaging and analysis. As for the expansion beyond the target identification, so basically previously most of HDS are applied for target identification, but now it's no longer just limited to that, but also applies to various stages of drug discovery and development, include not limited to toxicology studies, formulation optimization, and personalized medicine. There's also the increase in outsourcing to CROs, and CRO stands for the contract research organizations. So basically now there's a growing trend of pharma companies outsourcing this kind of HDS services to those research organizations. The reason why is that, first of all, it's more cost effective. And then second, it has greater access to the specialized expertise. It's more flexible, and then it allows the companies to focus more on their core competencies. And then, so these are the several leading companies in the HDS markets that I found. And then here is a table showing like top 5 companies with their key products, services, recent innovations, and market strategies. Yeah, as you can see here. So among these 5, 3 of them are actually based in US. There's one based in Germany, which is the Merck KGaA. That's a global provider of HDS products. Yeah, we can pause here a little bit just to read through the table. But lastly, so for some of the key takeaways, we're really seeing a rapid growth, market growth, primarily fueled by the technological advancements and increasing R&D investments. The global HTS market, valued at around $22.1 billion in 2022, now is projected to about 15 million by 2032. And secondly, there's a dominance of the cell-based assays and software services segments. So that's value. The cell-based assays basically is valued at around $12.2 billion in 2022, and prefer for the ability to generate more complex biologically relevant data. And geographically, North America continues to lead the market, expected to growth at around 7.7% by 2032. But we also see the significant emerging opportunities in the Asia Pacific region, driven by increasing the R&D investment there and the growing biotechnology sector. And then so lastly, it's clear that innovation and strategic partnerships are the key to success in this competitive landscape. Companies that invest in cutting-edge technologies like AI integration and 3D cell cultures, while also forming strategic alliances, are the best positioned to capitalize on the market's growth. And that's it.
Okay, thank you everyone. That's it for this journal club, and we hope to see you on the next one.